This is my neuron. We have the dendrites represented by the skewers coming out of the cell body, and they receive messages. The cell body is represented by the green apple with the nucleus and nucleolus inside of the nucleus. The soma are also just the space in the cell body. The axon is the skewer on the inside right here, and the axon hillock is the part right there that comes out of the it's the first part of the axon that comes out of the cell body. The myelin sheath are represented by the cardboard right here. They protect the axon. The nodes of Ranvier are the spaces in between each cell right there. The terminal branches and synaptic terminals are the paper right here. Neurotransmitters are in the synaptic terminals, and they get released into the synapse. And the sodium and potassium ions are inside the axon. And as I said, the axon is this skewer, but there's not enough space for the ions to be represented in there. So I'm going to pretend like the axon is the inside of the cardboard. So the sodium cells are the green M&Ms, and they're outside of the cell, resting potential, and the outside is positively charged, whereas the potassium ions, which are the orange M&Ms, they are inside of the cell, resting potential, and they are negatively charged. So first, resting potential. Basically, it just looks like this. It's normal neuron. The neuron is at rest. The outside of the cell has an excess of sodium ions. The inside has an excess of potassium ions. The inside is negatively charged, whereas the outside is positively charged. Depolarization of dendrite. So up here, so some of the sodium ion channels open and some sodium ions move into the cell at the dendrites, depolarizing them. So if the sodium ions were out here, they would move into the cell through the sodium ion channels, and that would depolarize it in the dendrite. And then the neuron reaching the threshold. The neuron has to reach a threshold level in which the excitatory signals have to exceed the inhibitory signals by a certain margin. If this threshold is reached, an action potential is guaranteed. So by the sodium ions moving into the cell, that depolarizes it, which gives it a more positive action potential, and if it reaches a certain level, an action potential is guaranteed. Depolarization of the neuron through the axon. If the threshold level is reached, all sodium ion channels open. So all of the sodium ion channels would open, and the sodium ions would flood into the axon cell down here, and this would make the inside of the cell now positive and the outside negative, which would depolarize it. Once this happens, the adjacent parts of the membrane become stimulated, and they begin this process as well, and the action potential continues along the axon to the synaptic terminals. So they go from the cell body down to the axon, to the adjacent cells, all the way down to the synaptic terminals down here. And that would be the action potential electric form in the axon. Release of neurotransmitters down here. The neurotransmitters are the blue M&Ms, if we remember. The neurotransmitters in the synaptic terminals are released into the synapse, which would be the space in between each neuron. The neurotransmitters then bind with the receptors on the receiving neuron and open them momentarily, allowing atoms to flow into the membrane. The atoms are either excitatory or inhibitory, and the excitatory cells or excitatory signals start a neuron's action potential, whereas inhibitory signals stop, slow down, or even prevent the action potential from happening. Reuptake or destruction of neurotransmitters by enzymes. The neurotransmitters left over in the synapse, so if they're still in here after the action potential is finished, they're either broken down by enzymes and given off as waste out of the body, or they are pumped back into the sending synaptic terminals for further use. So if one of these M&Ms was out here, it would either be thrown out as waste, or it would be pumped back into these synaptic terminals for further use. And that is my how to build a neuron project.